Hello everyone, welcome back to my channel. Today, when I stepped into my backyard, I found this small puddle of water near a leaking pipe. This water has been sitting here for quite a long time, completely stagnant. This makes it a perfect place for microorganisms to grow. I used this glass jar to collect some sample. After collecting the water from the puddle, I noticed it was surprisingly clear. That usually means most of the microbial life is not floating freely, but hiding around the edges. So I collected some of the green algae using a stick. And then I also picked up a few plant roots and some of the surrounding mud. Roots and mud are rich in nutrients and are one of the best places for microorganisms to grow and interact. I am adding all of this into the same glass jar, because the mixed sample gives a much better picture of life in stagnant water. I left this jar undisturbed for some time, allowing everything to settle down naturally. This is my garden microworld jar, started today, that is 28th of December 2025. As you can see, the heavier sediments have settled at the bottom. Mud, decaying plant matter, and moss. The algae is slowly spreading and the plant roots are still clearly visible inside. This settling process is important because it creates different microenvironments in a single jar. Oxygen rich zones near the top and nutrient rich zones near the bottom. I opened the jar without disturbing the layers too much. The green material you see is mostly algae and moss, and this is where a lot of microscopic life usually gets concentrated. Using the forceps, I am taking just a very small amount from the bottom. You don't need much. Even a tiny piece like this can contain thousands of microorganisms. I am now squeezing this sample onto a clean glass slide. You will notice there is water from the sample on the glass slide. So I am gently spreading it out. This helps the organisms stay alive and move naturally. Now I am placing the cover glass over the sample. I am using two forceps to lower it slowly, so the glass touches the water from one side first. This method helps push the air out, preventing bubbles from getting trapped inside. Air bubbles can distort what we see under the microscope, and they can also disturb the microorganisms. By lowering the cover slip gently and evenly, the sample stays spread out and undamaged. This step makes a big difference. Let's begin with the fast moving microorganisms from this garden sample. The first organism you are seeing here is called Allosoma. At first glance, it looks like a tiny yellowish worm. But what immediately stands out is how quickly it moves. It doesn't stay still for long. Instead, it glides, bends, and changes direction smoothly across the field of view. Following this organism under the microscope is not as easy as it looks. Just when you think it has slowed down, it stretches forward and moves away again. Allosoma is a microscopic freshwater worm, commonly found in ponds, puddles, and algae-rich water. Its fast, flexible movement makes it a perfect organism to start the fast movers category. Even at this zoom level, you can clearly see that it's actively exploring its surroundings, constantly in motion. Now as we zoom in closer, the movement of Allosoma becomes even more interesting. The body is soft, flexible, and slightly segmented, allowing it to stretch and contract rapidly. The yellowish color comes from its internal contents, which makes it stand out clearly under the microscope. Watch how the front end extends forward, anchors briefly, and then rest of the body follows. This repeated expansion and contraction is what allows Allosoma to move efficiently. Unlike organisms that swim using hairs or fin, this worm relies entirely on its flexible body. That simple design makes it fast, adaptable, and capable of squeezing through very tight spaces. The next organism is called Philodina megalotrocha, a type of rotifer. At first glance, it may look like the organism is not moving much, and that's actually true. The body itself is mostly stationary here. But if you look carefully near the front, you'll notice something very interesting. Even though the rotifer isn't swimming around, it is extremely active in another way. The wheel-like structures near its mouth are moving continuously. These tiny hair-like parts are spinning so fast that they create visible movement in the surrounding water. Even at this zoom level, you can already see small particles in the water shifting direction. That tells us the organism is actively feeding even while staying in one place. This is an important observation. Not all microorganisms need to move the entire body to survive. Some of them stay anchored and let the feeding system do the work. Now, as we zoom in much closer, the real beauty of the organism becomes clear. The wheel-like motion you are seeing near the mouth is made up of thousands of tiny hair-like structures. They move in perfect coordination, creating strong water currents around the rotifer. The organism itself is moving very slowly, almost staying in the same position, but the feeding structures never stop. Watch closely at the water around the mouth. You can see particles being pulled inward, almost like they are caught in an invisible flow. This is how this rotifer feeds. Instead of chasing food, it brings the food itself. The spinning motion pushes water outward on one side and pulls water inward on the other. As a result, food particles are guided directly toward the mouth. What's fascinating here is that the water itself becomes visible through motion. Normally, we can't see water move, but because of these tiny structures, the movement of water and particles becomes very clear. This organism may appear simple, but this feeding mechanism is extremely efficient. It allows the rotifer to stay in one place, save energy, and still collect everything it needs to survive. Even at this microscopic scale, you can clearly see intention and function. Nothing here is random. Every move has a purpose. This wheel-like feeding system is actually the reason rotifers get their name. Under the microscope, it looks like rotating wheels at the front of the body. All of this activity, the spinning, the water flow, the particle movement, is happening in a drop of garden water. This is Philodina megatrolocha, not racing through the slide but patiently feeding, shaping the water around it, and showing us how much life exists in places we usually ignore. Next in the fast movers category is an organism from the genus Chaetonotus. This organism moves extremely fast across the slide. It doesn't stay in one place at all, and following it under the microscope feels like a constant chase. Just when it comes into focus, it changes direction and quickly moves out of view, making it difficult to observe for long. If you look closely at its body shape, this organism has a long flattened form that helps it glide smoothly through the water and along the surface of the slide. Along the body, you can notice tiny spine-like or hair-like structures giving it a slightly rough outline. At the very end of the body, there are two clear tail fin-like structures. These act almost like anchors or stabilizers, helping the organism grip the surface and change direction quickly. Because of this body design, its movement looks more like gliding than swimming. The shape, surface structures, and tail fins all work together to make it fast, agile, and extremely hard to track. Here we have a nematode, one of the most common and fascinating microorganisms in garden water. Look at its long, slender body. It's smooth and flexible, tapering at both ends, which allows it to wiggle and glide effortlessly through the water. Even though it may appear simple, watch closely how it moves. The body bends and curves in a wave-like motion, allowing it to navigate and change direction quickly. Next we have Lepidella, another fascinating rotifer from the garden jar. At the zoom level, you can see its entire body clearly. It has a distinct, sharp tail at the back, which helps it anchor and maneuver in the water. You can notice subtle movements as it adjusts its position. Even from a distance, the tail is clearly visible, pointing downward like a tiny anchor. This structure is one of the key features that makes Lepidella easy to identify under the microscope. Watching this organism is a reminder that even when stationary, microscopic life is perfectly adapted to survive and interact with its surroundings. Now, as we zoom in closer, the body structure of Lepidella becomes even more fascinating. You can see the fine details of its feeding apparatus, and the sharp tail at the rear is prominent. It is used for stability and movement control. What makes this scene even more interesting is that nearby we can spot diatoms moving smoothly. The elongated bodies glide through the water effortlessly, creating a subtle dynamic in this tiny ecosystem. Lepidella and the diatoms together show how different microorganisms coexist in a single drop of water. The contrast is striking. One organism mostly anchored with delicate feeding movements, and the others gliding past, exploring freely. Now we are moving into a completely different world, the world of slow movers. What you are seeing here is a diatom. If rotifers and nematodes are like tiny animals, diatoms are more like microscopic plants encased in glass. Diatoms are single cell algae with beautifully patterned shells made of silica, almost like tiny pieces of natural glass art. These shells, called frustules, come in all kinds of shapes and designs: circles, ovals, boards, diamonds, and even star like forms that are truly breathtaking to look at. These shapes aren't random, they reflect differences in how each species live and how it interacts with its environment. It's like every diatom has its own architectural style etched into the silica of its shell. You might be wondering how something encased in glass can move, and that's a great question. Most diatoms do not have flagella or cilia like some other microorganisms, so they don't swim the way silicates or rotifers do. But some diatoms, especially those with a long slit like structure called a raphe, can glide slowly across surfaces. They do this by secreting a sticky mucus substance through the tiny slit, which grips the surface and lets them move inch forward in a way that almost looks like smooth sliding. Under this closer view, the motion you are seeing isn't frantic like a swimming protozoan. It is deliberate and calm. The diatom doesn't whip around, but it shifts, pauses, and slides almost like it's feeling its way across the surface. You may also notice subtle changes in direction, sometimes slightly rotating, as though it's scanning its surrounding. Remember, diatoms are photosynthetic. They use sunlight to make food just like plants do.
Their golden brown pigments capture light, and that's part of why they often appear with a warm glow under the microscope. So, in a way, they are tiny solar powered machines, beautifully armored, able to glide when they need to reposition, and constantly converting energy from light into life. What makes diatoms fascinating is not just their shapes or movement, but the fact that they are everywhere, from ponds to ocean, from moss to puddles like yours. In nature, they are some of the most abundant organisms on earth and produce a huge amount of oxygen we breathe. What you are seeing here is Spirogyra, a type of green algae commonly found in fresh water. At first glance, it looks like a long green thread or rod, almost like a microscopic strand floating in water. If you observe closely, this filament is made up of many connected cells joined end to end, forming a smooth, continuous structure. The bright green color comes from chlorophyll, which means this organism uses light to make its own food. Spirogyra doesn't move quickly like animals do. Instead, it drifts gently. This is a bright field view under 50x. You can see a rotifer moving at the below. And I think this is a paramecium. You can also see a ciliate close to the paramecium. Look at the way it's moving. Actually, it's difficult trying to keep it in the center. That's one more paramecium. I think that's a paramecium. Kindly let me know if you know what's this microorganism. Both are looking similar, but one is moving slower and another faster. One more rotifer in the center. Yeah, paramecium, the green color. You can also see a nematode below. Yeah, this is under dark field. Yeah, one rotifer coming to the left and one more. I think that is a paramecium. Yeah, two nematodes now clearly visible. This looks like a ciliate. I'm just panning through the slide, trying to explore all the places. Yeah, one more paramecium here, and you can see the ciliates also in the background. Yeah, nematode coming up. I think this is paramecium, but I'm still confused. Yeah, lot of nematodes. I'm still trying to find a tardigrade, but I'm not able to. This is a worm in the center. This is a confusion which I am having right now. Kindly let me know in the comment section if you understand whether this is a paramecium or it is a different organism. Look at the way it's moving. You can see the cell organelles also, but I'm not sure whether it is still a paramecium. Please do like if you enjoy this video, and kindly subscribe if you want to follow more microscope videos. You can see how the diatoms getting disturbed by this movement. Again, the same organism in bright field, in a different place. I'm just trying to look at its features, but it's getting hidden. Yeah, I can see the hair-like structures clearly now. You can also see the movements. Please do like, share and subscribe. Thank you.